So that brings us to Frank Graham, who is a professor at McMaster University. And he developed a cell line. Now, these cell lines turned out to be very important. The cell line that he developed was called HEC293. And these cell lines are used to grow the adenovirus vaccine, which is used for the viral vector types of vaccines. So what is a cell line, and how is it used to make a non-replicating virus? So first of all, we start off with cells. And in this particular case, these HEC293 cells, the HEK stands for human embryonic kidney cells. And you may have heard of some controversy about using these human-derived cells for these experiments. And there's no doubt that it is controversial, and I'll mention a little more about the ethics behind this in a few moments. But what happens is you take these cells and you can, a kidney, and you can take the cells from it and you put the cells in a petri dish and try to grow them. And they'll grow for about 10 days or so, but then they will die. But what was found, or what was known by this 1970s, was that some cells didn't die, especially if they had been infected by a virus. Then those cells became immortalized. And of course, that's what a cancer is. It's a cell that just doesn't die. It just keeps on growing and growing. So what he was trying to do was to immortalize these cells by mixing viral DNA, and the DNA from the virus of the adenovirus. So he mixed it with this DNA, and then he kept on growing these cells. And then he found after a certain period of time that some of those cells had become immortal, and that even after 100 days, they were still living. And in fact, some of those cells were expressing some of the information that was encoded in that viral DNA. And this is where it gets a little bit more complicated. You can take those viruses and you remove certain genes from them, and so they won't grow in normal healthy cells, but those viruses will grow in these immortal cells because they have the missing genes. And the space for the missing genes is replaced with the gene such as the spike protein gene from the SARS-CoV-2. And I'll go through that again in the next slide just to show you how that actually happens. But before I leave this slide, I just want to tell you about this book about the ethics of using human cell lines. And so there's a really great book called The Immortal Life of Henrietta Lacks. And she was the first person to have her cells taken from her. She had cervical cancer. It was around, I think, the 1940s or so. And the physician who was treating her took some of the cells from her cervical cancer and put them in a petri dish and found that they just kept on growing. And that cell line has been used over the decades for lots of research. It's been very beneficial to science. But the question all came out about, well, what are Henrietta Lacks' rights to her very own cells, and what about her family's rights to these cells? So it makes it a very interesting read, and you learn about some of the laws that have been put in place to protect people. So how does this cell line work in producing an adenovirus that is non-replicating? And if the adenovirus is non-replicating, how can you grow it in the first place? So what happens is you create an adenovirus that is missing several of the genes that are needed for replication. And then you infect a HEC293 cell, and the virus goes in and it produces its messenger RNA, it synthesizes the proteins, and those proteins begin to assemble back into a new virus, but it's missing some of the parts and it can't reassemble, except that some of those missing genes are supplied in the HEC293 cells. So now this virus can assemble and it gets released, and now you have these more copies of this adenovirus vaccine that can be used for a vaccine. And so when you inject a healthy person with these viruses, the adenovirus can't replicate. It can produce the the spike protein, but it can't reassemble and form new viruses. So that's the advantage of that type of technology, that the virus can't grow in you, but it can produce that protein, and then your immune system can respond to that spike protein. So this is the technology that's used for a number of vaccines: the AstraZeneca vaccine, the Sputnik vaccine from Russia, the Johnson & Johnson vaccine, the CanSino vaccine. That was a joint project between Canada and China, but which got scuttled with the current ongoing problems between Canada and China. These vaccines appear to be a little more reactogenic than other vaccines, but they haven't really published all the information on it. It appears to have an increased risk of blood clots and stroke in women between the ages of 25
and 55, with a 5 times increased risk.
And it has efficacy in the range from 65% to 91%. Now, the last type of vaccine that I'm going to talk to you about is the messenger RNA vaccines. And if you trace back these types of vaccines, the very first reported invention of it was way back in 1978. And that's when Georgios Dimitriadis found that if you put messenger RNA inside a tiny droplet of fat, and when you injected that droplet into a mouse, the droplets were taken up by white blood cells, which then translated, they produced that coded protein, and you created an immune response against that protein. Now, interestingly enough, this graph here shows you the citations, the people who referred to that original paper, and you'll see that there's only 26 citations. And so basically, Giorgio Dimitriadis's work disappeared. No one knew what he had done that long ago. It's only recently that people have rediscovered that he was the original inventor of this technology. And I looked for a photograph or the history of whatever happened to Georgios Dimitriadis, and I couldn't find anything about him. I don't know actually where he's gone to today. Here's how this particular technology works. And I had to put together two different diagrams, one from the CDC and one from the Moderna website, because I found that no one really had a good picture of what's going on here. So on the left-hand side here, we have this SARS-CoV-2 virus, and inside it we have the messenger RNA. And we take that messenger RNA sequence that codes for the spike protein, and we modify it a little bit. So normally, if that messenger RNA got inside one of your cells, your body would almost immediately recognize it as being forward and start having an immune response against it before it got very far. But one of the tricks is to actually get rid of those foreign signals. And so it makes this messenger RNA kind of stealthy. And that allows the messenger RNA to act without being detected. Now, if you had just injected that messenger RNA directly, you have enzymes in your body that would immediately digest that messenger RNA and it would be gone. So you need to protect it a little bit, and that's where this little fat droplet comes in. And so the messenger RNA is packed inside this lipid nanoparticle. And that lipid nanoparticle is a composition of positively charged lipid, lecithin, cholesterol, and polyethylene glycol. This formulation has been worked on over the last 10 years to really refine this to come up with this sort of optimum formulation of this little lipid particle. You'll see that they're about 80 nanometers wide. So that's that it's that same virus-like particle size where it gets picked up automatically by your cells and recognized as something like a virus. So when you inject these little lipid nanoparticles can be picked up by either muscle cells or by specialized cells from your immune system. And then the messenger RNA gets directly into your cytoplasm. And here are those little machines, those little ribosomes, and they take the message in the messenger RNA and they encode the protein. And then that protein is what your immune system responds against. And so now you've created a strong immune response against those proteins. And of course, the advantage of producing that inside the cell is because that's how a virus would do it. And so your immune system recognizes that it's an invader coming from inside, and a whole branch of your immune system that responds to that kind of infection is then activated. And so this particular technology is for the Pfizer vaccine and the Moderna vaccine, and also CureVac. And actually, there's a company in Canada called Providence Therapeutics. I remember they were on the radio last summer begging the government for money. They didn't get it until finally in the fall. Why there was the delay, who knows? And there have been some allergic reactions associated with the Pfizer and Moderna vaccines, which are licensed, but it's quite rare, 5 cases in about 1 million, and it tends to happen in people who have known allergic reactions. And so that's why you have to wait around for about 15 minutes after you get it to make sure there's no allergic reaction, and it can be treated, of course, with an EpiPen. And the efficacy of these vaccines is quite high, about 95%. So putting this all together now at the end, this is a coming diagram from our publication. How do the different vaccines compare? And so, we looked at these different vaccines, and we put most of the emphasis on the efficacy. I think most people want to see a very high efficacy, but also the safety, the reactogenicity, that's also an important feature, and then the logistics and the cost of manufacturing them. And we rank these vaccines, and this is the ranking that we came out with. So the messenger RNA at 82, then the protein vaccines at 80, the viral vectors at 74, and the inactivated virus ones down at 71. And I just want to point out to you that these different vaccine manufacturing technologies do have some differences, but all of them are very good. They all have very good safety profiles. They will all protect you. And so I suggest that any vaccine that you're offered, you should accept it because you will get some protection.
Here is a link to the publication, and it's open access, so you can access it online.
And that is the end of my talk. Thank you very much for your interest and attention.
And remember, with a vaccine, you get to choose which one and when.